Good morning, everyone. It's my great pleasure to be here to share this talk. The topic is about scaling fermentation with precision, especially from the perspective of process development to industrial biomanufacturing. So there are significant challenges when we scale up fermentation processes. Variable factors influencing fermentation performance and product outputs. So one factor which is particular to aerobic fermentation, which is oxygen transfer limitation. The other, which is common to aerobic fermentation and anaerobic fermentation, nutrients gradients, fluid dynamics, temperature, pH gradients, etc. So all these factors can adversely affect cell performance, process stability. As a result, it can have low product yields, productivity, and compromised product quality. So it's very important to understand key variables affecting fermentation performance. Some approaches we can adopt like data-driven approaches and tightly controlled process development as well as stream engineering, aiming to mitigate risks. And the importance of scalable fermentation, we use fermentation at larger scale to support reliable production of essential medicine, enzymes, specialty chemicals, biofuels. So it can also contribute to environmental friendliness, promoting sustainable production technologies for healthier planet. So the innovation in the fermentation or biomanufacturing system can advance in biotechnology, material science, and chemical industry, etc. So the key pathway for scaling up start with a streamlined stream selection, which is very important, which can facilitate the next stages for high product yield, productivity, and titer. So next step is media optimization, not only for cell growth, but also for fermentation media. So it's also necessary to explore the novel formulation for the fermentation, especially at the larger scale. And next stage after that is the pilot run. So it's important to conduct pilot runs to minimize risks and also provide insights before larger scale. And final stage is industrial production, which is a transition from pre-pilot scale, pilot scale, demonstration scale, pre-commercialization scale to full scale. So at the full scale, it's very important to ensure product quality and economics of scale. So in our lab, we start with a small scale, and we also did pilot scale for benchmarking. So we work on 3 major categories of fermentation products. Chemicals as examples of extracellular products. Another one is incellular products represented by PHA, which is biodegradable polymers. And there is one more category, which is living organism, which is probiotics. And in the following couple of slides, I will talk about our research from lab scale to pilot scale, especially related to PHA fermentation. Dr. Yue also mentioned PHA fermentation, so I'm going to share more about what we are working on in this topic. So we are really motivated to synthesize biodegradable plastics. There are tremendous issues to address plastic pollution. So traditional way, take, make, use, dispose. At the end, plastic pollution arises. And there are, so now the emerging way how to address, plastic pollution from how to synthesize biodegradable plastic and also how to recycle waste plastics. So we actually work on both. So in the next slides, I'm going to show two cases in this topic. And first one is how to make biodegradable plastics, which is PHA, from waste carbon source, particularly food waste. So we use waste cooking oil as a feedstock, and here, and we produce PHA at the end. We start with a flask culture I didn't show here, which is 2 liter, and further scale to 10 liter, and all the way to 130 liter reactor with a working volume 70 liter. So here is the fermentation broth discharged from this pre-pilot fermenter. And as I mentioned, here is cells, and here is media chain PHA extracted from the cells. So this is a good example about how we scale up from a bench top to a pilot scale or pre-pilot scale. And next one is about P2P, plastic waste to plastic. And we use the post-consumer plastic waste. And we collaborate with Dr. Bai at Iowa State for plasma deconstruction to yield fermentable intermediates, which is called OI. The full name is oxygenated intermediates and liquid. And after making emulsion, so we do flask culture. So we optimize the process. We use the co-culture together with a fed batch. And we are able to scale to 2 liter. So next stage will be a big scale. So this is a fermentation broth from the 2 liter flask. So here's the cell from this process. So again, the plastic waste is substrate. And here is the final product extracted from the cells. So we are also working on the downstream processing in terms of PHA extraction. In addition to this project, we are also working on specialty chemical as a fermentation product and the living organism, which is represented by probiotics for the fermentation system. So expect outcomes from today's talk and the showcase activities this afternoon. So it's understand the process robustness challenge in the scale up, and also learn process control and analytics, how to control and predict the process stability. And also, as I mentioned, there are different categories of fermentation products, so we should come up with different design strategies for maximize the fermentation system performance. So the future direction is AI, machine learning for the predictive process control, for tighter yield productivity, high throughput automation, accelerating strain development, and iterative testing cycle, synthetic biology integration with advanced sensor networks. Digital twins are also powerful, which can provide virtual bioreactor models for de-risking at a small scale before we scale up to large scale. So overall, we are aiming to provide a platform toward a resilient bioeconomy, which is transforming research into reproducible industrial solutions. So my main expertise is about process development. So I'm very open to discussion and collaboration opportunities, for the digital twins, AI, digital, manufacturing systems development. So with this slide, I'm going to conclude today's talk. So I would like to invite to our showcase this afternoon. So through this showcase activity, I hope we can share, the fermentation process in the bioreactor. We will also have that one in our exhibit. Due to location constraints, I'm not able to show live bioreactor operation, but actually we are doing that in our lab. We will walk you through the fermentation process. We will also share the samples, including substrate and fermentation products with you. So with this, I'm going to conclude my talk today. Thank you.